Hello, my name is Kathleen Loftus, and my IS explored how mothers influence the gut microbiomes of their infants. I am the first College of Wooster biology major selected to conduct my research in the form of a literature review. My interest in microbes began after studying the oral microbiome as part of my study abroad program. Following my junior year, I worked as a clinical research intern in the maternal fetal medicine department at Metro Health. The experiences I have had through both of these opportunities has helped to shape the topic chosen for my literature review. Microbes are universal and inhabit nearly every environment around us, yet there is a core microbiome found within and throughout each host. During the first days of life, organisms come into contact with and are colonized by an array of different microbiota. These commensal bacteria aid in immune function, nutrient processing, and other metabolic processes of the host. Our gut microbiome assists in the metabolic breakdown and synthesis of several substances important for daily function and survival. Therefore, overlapping abundances of bacterial species of the core gut microbiome, such as firmicutes and bacterioids, are integral for proper development and daily function. The timing of gut microbiome establishment in early life influences brain function, behavior, and stress responses. The infant gut microbiota is highly dynamic, unstable, and exhibits low diversity before it shifts into a more stable, adult-like pattern over the first year of life. Proper microbial colonization and stabilization is vital for proper development of the body's communication networks, such as the HPA axis. The HPA axis is the major regulatory system in processing how the body responds to stress. The HPA axis relays information via the neuroendocrine system in order to maintain homeostasis during and after a stress response. Gut microbes are part of this network as they help in building resistance to changing external environments that may cause stress and anxiety. The gut-brain axis involves biochemical communication between the gastrointestinal tract and the nervous system, which is linked to emotional and cognitive centers of the brain. Bidirectional communication between the brain and gut has been established as these systems relay signals through the nervous system, neuroendocrine system, and immune system. Changes in microbial composition are linked to changes in adipose levels and energy balance, intestinal permeability, inflammation, and increased levels of stress, anxiety, and depressive symptoms. Investigating the stages of microbial colonization and development via maternal influence offers insight in better understanding the role of microbes in our bodies. Maternal health factors and actions such as drug intake are well known to have detrimental effects on the development of the fetus. Less well known are the influences prenatal factors such as maternal stress, diet, weight, vaginal infection, and antibiotic use can have on the infant's gut microbiome. While the long-withstanding sterile womb hypothesis has dominated scientific thought since the 19th century, a recent shift in understanding has spurred further investigation into what gets transferred to the fetus while developing in the womb and the long-term effects of the maternal-fetal relationship. The sterile womb hypothesis claims that the in utero environment provides a barrier that keeps microbes from reaching the developing fetus. The following assumption was that any microbes found on and in newborns must be obtained during vaginal delivery or from the surrounding environment. Further support for the sterile womb hypothesis has stemmed from the association between the presence of microbes in the fetal environment with neonatal infection and other threats to the pregnancy. In utero transfer is hypothesized to occur when microbes from the mother, most likely from the mouth or gut, are transferred to the infant while in the womb and supposedly assist in colonization and development prior to birth. Support for this hypothesis stems from more recent studies finding and building on detection of bacteria throughout different areas of the womb. The in utero transfer hypothesis proposes that while the fetus develops in an environment protected from pathogenic bacteria, selective maternal microbes still pass this barrier. Maternal stress has also been found to have an effect on the infant gut microbiome. Prenatally stressed monkeys have offspring with altered intestinal microflora characterized by lower levels of both bifidobacteria and lactobacilli. Other studies on prenatally stressed mice suggest that prenatal stress may have the ability to alter the composition and functioning of the pup's microbiome for an extended period of time. Mothers of infants that underwent high stress during pregnancy also had altered gastrointestinal bacterial abundances. Intrauterine infection is strongly correlated with preterm labor, which implies the ability of microbes to influence signaling pathways. Rupture of membranes is also associated with particular bacterial species. While there is evidence of prenatal antibiotic exposure affecting specific gut microbes, there are also implications for larger microbial shifts in the gut. A group of infants exposed to antibiotics prenatally had bacterial community levels associated with dysbiosis and possible increases in necrotizing enterocolitis occurrences. Choosing between the risks of foregoing antibiotics and the possible risks of antibiotics hindering healthy fetal development is a decision that mothers have to weigh. Infant gut microbial establishment is influenced by maternal weight and weight gain during pregnancy. Evidence of prenatal prebiotic and probiotic administration alleviating children's increased risk of obesity connected to maternal obesity during pregnancy is continuing to grow. Cesarean section and vaginal delivery are both common routes of birth in humans, and evidence of differences in the gut microbiome of infants born these two ways exists. Since early life, microbial makeup has significant influence over the long-term health and development of the infant. Understanding factors that may alter colonization could help to improve the field of healthcare. A longitudinal study found that levels of lactobacilli in the vaginal microbiota of pregnant women are higher than those in non-pregnant women. This bacterial shift that spans across mothers during pregnancy suggests the evolutionary importance of these microbes for the developing infant. Therefore, the primary source of lactobacilli in the infant gut most likely comes from contact with vaginal microbes during birth. RNA sequencing of meconium, which is the first bowel movement of a newborn, obtained 24 to 48 hours after delivery from healthy full-term infants, found that cesarean section infants had significantly lower occurrence and diversity of lactobacillus species when compared to vaginally delivered infants. Continued monitoring in this study found that the differences in lactobacillus and L-Gasseri levels between vaginal and cesarean infants persisted through 6 months post-birth. Therefore, differences found in the meconium of C-section infants may be linked to early priming for gut dysbiosis. Cesarean deliveries are also associated with metabolic disorders in infants. C-section babies at 1 month old have an overgrowth of C. difficile and are prone to increased body mass and childhood obesity. These microbial differences between vaginal and C-section infants are indicative of larger and long-term health implications. Research maintains that neonatal microbiota develop optimally in full-term vaginally delivered, breastfed, and non-antibiotic treated infants. The evolutionary implications of vaginal delivery are underscored as maternal transfer of microbes spans across an array of invertebrates and vertebrates. Invertebrates often perform egg smearing, in which the mother coats her eggs in microbes, or passes a fecal pellet that is consumed by the larvae immediately following hatching. Turtles are one example of vertebrates exhibiting maternal vaginal transfer. Several bacterial species have been detected from the cloacal opening in green turtles immediately after egg laying. Other bacteria found in the eggs of river turtles are most likely transmitted from the mother, since these species are not found in their environment. Since all human babies were born via vaginal delivery until about 100 years ago, the vaginal birth route is hypothesized as an evolutionarily important mode of microbial transmission associated with proper offspring development. As a precautionary measure for infants not naturally exposed to vaginal microbes during birth, A possible intervention method for cesarean infants involves microbial swabbing. This process is performed by using a Q-tip to transfer vaginal microbes all over the newborn's body immediately following birth. Since humans have been delivered vaginally for centuries prior to modern medicine, lacking this natural exposure to the vaginal microbiome may be linked to health problems. The short-term effectiveness of swabbing is supported by evidence, yet more longitudinal research needs to be done to better determine the persistence of microbial differences between vaginal and C-section infants and to track other possible health implications connected to birth route. The average U.S. child will receive 3 courses of antibiotics by the age of 2. Antibiotics can have adverse effects on the sensitive developing microbial communities in infants in particular. The ability of antibiotics to directly alter the intestinal microbiota is well established, yet long-term effects on physiology and development are lesser known. Breast milk is packed full of microbes. The core microbiome of human breast milk contains 100 to 600 species of bacteria. Human milk provides essential dietary fatty acids and metabolites that aid in the growth and development of breastfed infants. The enteromammary pathway assists in transferring maternal gut bacteria to the mammary glands through blood and lymph circulation. Following birth, breast milk provides factors that promote the growth of specific microbes that are essential for infant gut microbial development, such as bifidobacteria. Breast milk also contains microbes obtained from the outside environment, such as those from maternal skin and the infant oral cavity. The cessation of breastfeeding is required for maturation into adult-like intestinal microbiota, as opposed to the introduction of solid food. The finding that breastfed infants have lower levels of toxigenic bacteria suggests breastfeeding cesarean-born infants may assist in alleviating perturbed gut microbiota levels that are indicative of dysbiosis. Evidence is building behind probiotic benefits on breast milk via prenatal and postnatal administration. Maternal probiotic supplementation during breastfeeding holds potential in reducing risk of immune and metabolic diseases by providing microbes such as lactobacilli that assist in regulating different pathways throughout the body. Formula milk usually contains a mixture of cow or goat milk, vegetable oils, lactose, vitamins and minerals, and sometimes probiotics to provide protein, fat, carbohydrates, and beneficial microbes. A study following the development of infants for 2 years found that infant microbial disturbances were associated with antibiotic exposure, cesarean delivery, and formula feeding. Formula feeding disrupts the developing gut microbiome by altering colonization of maternal bacteria and microbial diversity and by delaying immune development. While formula feeding is often a necessary alternative to breastfeeding, research largely supports the importance of breast milk in transferring maternal microbes to infants that are vital for proper development. Prebiotics have the potential to improve formula milk mixtures to further aid microbial administration. The human gut microbiome is constantly changing and evolving. Maternal influence over an infant's microbial composition prenatally, during birth, and postnatally marks a key period for microbial development and programming of bodily communication systems. Further research on the long-term effects of maternal influence on an infant's gut microbiome has the potential for introducing new gestational therapeutic interventions to help combat diseases and disorders associated with gut dysbiosis. I would like to thank Dr. Lynn and my friends and family for the constant support and encouragement. I would also like to thank you for taking the time to view my IS presentation.